video is the beginning of Unit 7 on genetics, learning target 1, and in this video we'll be talking about some of the different scientists that paved the way for the study of genetics and the study of genetics in its beginnings. First of all, we need a working definition of genetics. Genetics is the study of heredity and genes and how traits are passed from one generation to the next. And so you see here this picture, mom, dad have several offspring, or they just have one offspring, but that one offspring has chances to inherit different traits from mom and different traits from dad. And when we get to Mendelian genetics, we'll be talking about the specifics of those probabilities. But the point is that mom and dad can pass their traits on to their children. Their children will have similar characteristics to both mom and dad. Now, in the early 20th century, the general consensus in science was that proteins contained genetic material, and that proteins were what was passed from one generation to the next. And they did not believe the molecule of DNA. They just believed that a sequence of repeating nucleotides didn't have a lot of significance. And so basically what's going on in the early 20th century is with genetics is this idea of proving that DNA is the molecule that transfers genetic information. And so one of the first people to work with this is Frederick Griffith, and he was actually a doctor and was attempting to find a cure for pneumonia when he came upon another discovery. And his big claim to fame is he discovered the technique called bacterial transformation. Bacterial transformation. And this is the experiment that he did. So you need to know this, Griffith's experiment. Step 1 of the experiment, what he did was he took some bacteria that he knew caused disease, he knew caused the pneumonia disease, and he injected them into a mouse, and the mouse died with its leg in the air, apparently. So it dies of pneumonia, something he knew was going to happen. Step 2, he injects a mouse with harmless bacteria, and the mouse lives. So why would he do these two steps? What is the purpose of these two steps here? Well, we know in science that for any experiment, you need to control. These are variables that you know, that you control and you know the result of. And so he knew that when he put pneumonia in those mice, they were going to die, and he knew when they put the harmless bacteria in the other mice, they were going to live. And so next, we get to kind of the experimental part. The next step here, he's going to take some heat-killed, disease-causing bacteria. So he takes the bacteria that he knows causes pneumonia, and he boils them, he kills them. He injects them into mice. What happens? The mouse lives, because those bacteria are dead. They're no longer interacting with their environment. They're just dead. All right, so the next part, he takes the heat-killed bacteria that he knows cause disease. Of course, they didn't when they were dead. And he mixes them with live, harmless bacteria. So dead, harmful bacteria, live, harmless bacteria. And the result is the mouse dies. And so why does that happen? Well, he noted that the bacteria that he found, the living bacteria, had been changed. He did not know why they were changed, but he noted that there must have been some sort of transformation agent, something that caused the change. Now, this technique is used today to this bacterial transformation, causing bacteria to change, is used today for lots of applications, and we'll talk more about those later. But again, Griffith didn't know. He suspected that it might be DNA, but he would have been going across or against the general consensus of science. But what we know happened is that the live bacteria took up some of the DNA of the dead bacteria, and it caused them to change their behavior and become harmful. So that's Griffith's experiment. The next guy is Oswald Avery. And Oswald Avery, what he did was he studied and repeated Griffith's experiment, and with some different variables. What he did in Griffith's experiment is he isolated DNA, because his whole idea was to attempt to see if it was DNA that was this transformation agent that Griffith talked about. And so in one part of his experiment, part one, he removed the DNA from the cells, removed DNA. And those cells did not cause the disease. But in part two, he removed all but the DNA. So he just let the DNA by itself, and lo and behold, those cells caused the disease, killed the mouse. And so Avery seemed to have isolated DNA as the transformation agent, as the thing that is inherited from one generation to the next. But of course, science is slow in convincing, because it's a long-held belief that protein was the transformation agent. It's going to take some more convincing. And so that's where these two come in. Hershey and Chase, it's Alfred Hershey, Martha Chase, and they did their experiment in 1952. You don't need to know that, but that's just kind of a place marker in history. And their experiment conclusively proved that DNA was the change agent in organisms. That DNA is what's passed from one generation to the next and carries hereditary information. What did they do with their experiment? Well, they used something called a bacteriophage. This is a picture of a bacteriophage. It looks like an alien landing craft. And a bacteriophage is a virus that attacks bacteria. Why would they do this? Well, viruses, consequently, are made of two things: DNA and protein, both the molecules that are at hand here. And so they can use this bacteriophage to track DNA and to track protein and see which one ends up in the next generation. The way that viruses work is they inject their DNA into a host cell. So viruses inject their DNA into a host cell. That host cell takes up that DNA and then makes copies of it. Well, it just so happens that that DNA is what codes for the protein coding of the virus. And so essentially the host cell is making copies of that virus. So then the virus will eventually explode the cell. All those new viruses go out into other cells, and you have a viral infection. So they used radioactive tracers. They were going to attach radioactive tracers to both DNA and to protein in order to track which one goes into the next generation. So let me draw this. So in part one of their experiment, they're going to use radioactive sulfur, which is sulfur 35. Now, sulfur is not found in DNA, but it is found in proteins. And so what you end up having, if this is your normal looking bacteriophage here, all right, you're going to end up having bacteriophages that kind of look like this, with this nice protein glow-in-the-dark coating, because it's radioactive. Now the DNA on the inside is going to be normal. It's not going to be radioactive at all. And so these viruses then, draw some bacteria. Here's a bacteria. The virus kind of lands on the bacteria. It's then going to inject its DNA into the host. Well, that bacteria is going to replicate. And notice, in the next generation, there's no trace that that virus was ever there. There's no trace of those radioactive proteins. So that proves that protein is not being passed from one generation to the next. What are they going to do in part two? Well, same idea, except for this time they're going to use radioactive phosphorus 32. And because phosphorus is a major component of DNA. So the same idea, you can grab this guy. You're going to have a normal protein coating, but now you're going to have this radioactive DNA. It's really easy to trace. Easy to trace, think glow in the dark, if that helps. Alright, and so same idea as before. Here's your bacteria. Virus lands on the bacteria. Virus injects its DNA into the host. That cell's going to reproduce. And lo and behold, you have traces of that radioactive DNA in the next generation. And so what does this prove? DNA is what is being passed from one generation to the next. DNA is what contains the hereditary information. So Hershey Chase experiment, very important experiment in the formation of what we understand about DNA today. So real quick, just a couple of structural components of DNA. DNA is a nucleic acid. We know that. You can look at its name because Na stands for nucleic acid. And all nucleic acids are composed of 5 parts. It's a 5-carbon sugar. And in DNA, the 5-carbon sugar is called deoxyribose, which is where the D is. All right. A 5-carbon sugar, a nitrogenous base, which we'll get to shortly, what that is, and a phosphate group, hence the radioactive phosphorus. So all nucleic acids have those 3 things. DNA, its 5-carbon sugar is deoxyribose. And as for its nitrogenous bases, it has 4 of them. They are in 2 groups, purines and pyrimidines. The purines, here you can see they have 2 ring structures. 1, 2. The pyrimidines have just 1 ring structure. The 2 purines are adenine and guanine, or we'll just usually refer to them as A and G. And the 2 pyrimidines are cytosine and thymine, or C and T. And an easy way to remember this is you'll notice that the pyrimidines all have Y in them. One additional pyrimidine that you'll need to know that doesn't have a Y in it, sadly, is called uracil. And uracil is only found in RNA, which we'll get to in the next video. So that brings us to the next guy, Erwin Chargaff. First names are not important, but some of you may want to know his first name. Erwin Chargaff. Chargaff stated that the concentration of G is always equal to the concentration of C. And the concentration of A is always equal to the concentration of T. And so what that suggests is that A, adenine, only ever binds to thymine, and guanine only ever binds to cytosine, or a purine only ever binds to a pyrimidine, A with T, C with G. And if you know the concentration of one of those, you can then solve for the concentration of all 4. Let me demonstrate. So let's say that we know that a particular cell has 18% adenine in it. Well, we also know that it has 18% thymine based on Chargaff's rule, for a total of 36% A's and T's in the cell. That leaves us with what? 64% C's and G's. How to do that? It's not witchcraft. You just take 100 and you subtract 36 from it, and you get 64. Well, what do we do with this 64? We divide it in half. And there you go. So 18, if you know that you have 18% adenine, you can say that you know that you have 32% cytosine. Or you could just take 50 and subtract 18 from it and get the same answer. Either way. Almost there to the end of our study of DNA people. This lady named Rosalind Franklin, she used a technique known as X-ray diffraction. And she began to discover the helical nature of DNA. So her big claim to fame was that she discovered the helical nature of DNA. But before she could publish any of her work, and before she could go any steps further, she died of cancer at the age of 37. However, she did take this picture with her X-ray diffraction. You'll notice this picture is like down, if you're looking down the tube of DNA. Basically, DNA kind of looks like this. And if you were to like turn it towards you so you could look down the tube, almost like looking down a straw, when she did that, this is what she saw. This is like the outer rings of the DNA, or the outer rails. In the middle, you have those nitrogenous bases crossing. And so when she looked at this, she knew it was kind of like looking down a spiral staircase. So there's some sort of helical shape to the DNA, because the X in the middle denotes that spiral. All right, and she was almost on to the shape of DNA, but she didn't quite get there because she died. And so these two people picked up her work. They also used the work of Chargaff and put it all together, and finally came up with the idea that DNA is, this was 1953, just going to give you a heads up, DNA is or the shape of DNA is a double helix. They used 3D models to show this work. Their models are still extant today in museums, what they used. But they again discovered that DNA is a double helix. And what do we mean by double helix? It has this spiral shape that is double-stranded, and it kind of spirals in and out of each other, almost like a twisted up ladder. And the backbone, the red and green threads on this picture here, are made of sugar and phosphate, and the rungs of the ladder are made of nitrogenous bases. So what I'm going to do is I'm going to simplify this picture. You'll notice this picture is kind of flattened out, but there's lots of chemicals and things there, and a little bit too much for us. So I'm just going to simplify this. And essentially what you're looking at, when you're looking at a DNA molecule, what I'm drawing here, these little black dots are going to be the phosphate groups. These red blobs here are going to be the sugars, deoxyribose. Right? And then I want to draw some boxes to symbolize our nitrogenous bases. All right, and they don't have to alternate like that. There's always a purine with a pyrimidine, but you can have 14 pyrimidines in a row, but bound with a 14 purines on the other side. It doesn't really matter. But the important part of this drawing is what I want you to know is this sugar phosphate backbone, the vertical pieces of the DNA, are put together with covalent bonds. There's the phosphate, there's the sugar, and here you have nice, strong covalent bonds. Your cells do not split the DNA, or rarely split the DNA in half vertically. It takes a lot of energy. And so these bonds in the middle between the nitrogenous bases are slight attractions due to the polarity of those molecules. And what are these called? If you remember, these are called hydrogen bonds, which are very similar to the magnet on the refrigerator. Easy to pull apart, but will stick indefinitely if you need them to. And so just as a test here, if this was guanine, then this other side would be cytosine. And if this was thymine, this other side would be adenine. And we could just keep going down the line. So you kind of get the idea. C's and G's, A's and T's. And that's kind of what your average DNA molecule looks like.